My name is Kevin Rychel, and I recently finished my PhD at UC San Diego in the Systems Biology Research Group. In this video, I want to discuss my recent paper, which is the largest and longest paper of my PhD, so I'm really glad to finally have it out. You can read it in Cell Reports. It's called Laboratory Evolution, Transcriptomics and Modeling Reveal Mechanisms of Paraquat Tolerance. So let's jump right in. So the motivation for this study is really twofold. For one, we want to understand how cells can evolve in a laboratory to tolerate a large amount of stress. And the specific stress we're looking at here is called paraquat stress, which comes from a common herbicide that produces oxidative stress within cells. And we'll get a little bit more into paraquat later in this video. But we essentially use a technique called ALE, adaptive laboratory evolution, which exposes the cells to increasing amounts of this stressor over the period of months. And as the cells move from flask to flask through this ALE, they accumulate mutations in their DNA, which enable them to better tolerate more and more of this stress. And the second motivation for this study is actually to bring in a different perspective on these strains, which is coming from the RNA sequencing data. We want to understand how gene expression changes during an evolution like this. A lot of times with most ALE studies, you're only looking at the genome, the DNA changes, which are really informative and super interesting, but they don't cover all of the changes to these cells. And there's actually a lot of sensory information and really valuable strategies hidden away in the transcriptome. And with prior methods, it was just too much data. There's so many different differentially expressed genes that it can be really hard to understand exactly what is changing and what's important. But our lab has some expertise in this. We've developed iModulon analysis, which is a way to basically decrease the amount of variables and end up with these knowledge-enriched iModulons, which we can track, basically giving us the same information as differentially expressed genes, but in a much more interpretable, easier way. So again, we wanted to, one, understand how cells can learn to tolerate this stress, and two, we want to integrate iModulon analysis into ALE analysis and really start to make connections between the genome and the transcriptome. So I start this paper with some basic overviews, some categorization of the various changes and signals we observe. And I also show that 1,000% increase in the amount of paraquat that these cells can tolerate, which is a pretty dramatic improvement. And so then the next question is, of course, how did the cells become so tolerant? And that brings me to figure 2, which is my favorite figure. You can see 4 columns here. There's the changes to the DNA, the changes to the RNA, these iModulon signals that I mentioned, which again are much fewer and more easy to interpret now that we have iModulon analysis. And then we have metabolism and phenotypes. And so by drawing connections between all of these 4 things, we're really able to get a systems biology overview perspective on how all these different pieces work together to enable these cells to grow so well in this stressful environment. And at the bottom of the figure, I take you through a drawing of a cell that really brings it all together and shows you how it all relates to the way paraquat works. So you can see letter A here is near a transporter, and you can see paraquat coming into and also being pumped out of the cell. And in our evolved strains, we get an amplification of and an upregulation of this multi-drug efflux transporter, which is able to pump paraquat out. So that's one of the first clearest strategies that these cells are implementing, is just get it out. But the stuff that still gets in, the paraquat that does make it into the cell, is able to pass an electron off to oxygen and create these toxic reactive oxygen species, the oxidative stress agents, really. And so these, for one, the evolved cells are able to detoxify them a little faster because they upregulate some specific stress responses. And then also, one of the things that oxidative stress damages the most is iron and iron sulfur clusters. And we see some changes to the way iron is taken up and iron is regulated that ultimately enable the cell to feel less of that stress. We also see some changes to motility. A lot of the cells became more motile, which is interesting. I discussed that more in the paper. And then a lot of the signals, actually, most of the iModulon changes, most of the transcriptomic adaptations were just responding to the change in stress levels. The stressed out, unevolved strains were very unhappy. And you see after the evolution, a lot of those signals get turned down, and we really return to more of a homeostatic state. And then finally, what's really interesting here is paraquat doesn't just create that oxidative stress and stop. It actually continually produces oxidative stress by stealing electrons from the cell's energy-producing pathways. And this is a really major problem and why paraquat is such an effective herbicide. But we actually saw the evolved strains rewire their metabolism in order to avoid passing that electron off to paraquat, which I thought was a really exciting and interesting kind of complex change that occurred that involved multiple mutations and iModulon changes. So if you step back, you really see this systems level picture of all the different ways that paraquat has interacted with the cell and how evolution has turned all those things down. And all these mutations are really a goldmine of additional engineering and research that can be done. So there are three main impacts to this study. First, the specific mechanisms are really interesting, I think, for basic science. If you're a wet lab researcher and you're looking for hypotheses to validate around iron, oxidative stress, transcriptional regulation, this study really provides a lot of starting places for that type of research. Two, these mechanisms can be used to make cell factories work better. So a lot of times bioproduction, fermentation strains experience a lot of oxidative stress. And if you have one of these strains and it's affecting your yield, maybe you implement some of the changes that these evolved strains use. You add these mutations into your strains in order to allow them to produce whatever product you are making, produce that better. And then finally, the knowledge mapping method here, going from the genome and the iModulon changes and really relating between those two things, is a really novel and interesting systems biology perspective. And I've already used it in another paper to understand high temperature tolerance. And I think it can continue to be used really anytime you have two strains, a starting strain and a final strain, where the final strain has many systems biology adaptations that you can really understand from the perspective of the transcriptome. So with that, thank you so much for watching. Please check out this paper at Cell Reports, and please also go to imodulondb.org. You can learn a lot more there. Thanks again.